What is mitosis? Mitosis is the process by which a single cell divides into two identical daughter cells through asexual reproduction. Cell division starts with interphase and then moves into the 5 steps of mitosis. In prophase, the chromosome fibers condense and become more visible in microscope. The mitotic spindle begins to form from the centrosomes and microtubules. The centrosomes start to move away from each other. In this example of a cell, this cell contains 2 chromosomes. The DNA is duplicated and sister chromatids form within the cell. The next phase is prometaphase. In this phase, the nuclear envelope breaks away and the microtubules from the centrosomes spread out and the kinetochores form at the centromeres of the chromatids, where the microtubules attach. Next is metaphase. This is when the centrosomes are located at the opposite poles of the cell and chromosomes are lined up at the metaphase plate in the middle of the cell. Each sister chromatid is attached to a microtubule at its kinetochore from the opposite pole. After metaphase, anaphase occurs. The sister chromatids begin to separate and move along the microtubule, moving one chromatid to one pole and the other chromatid to the opposite pole. Then, in telophase, the nuclear envelope starts to develop at the 2 opposite sides. The chromosomes become less condensed and the microtubules go back to the centrosomes. Cytokinesis occurs after mitosis. This is when the cleavage furrow starts to form in an animal cell. The cleavage furrow eventually pinches the 2 animal cells apart. However, in a plant cell, this would occur by the formation of a cell plate between 2 nuclei instead of a cleavage furrow. The plate would eventually grow and reach the cell wall on each side, splitting into 2 daughter cells. What is meiosis? Meiosis is the cellular division of gametes and produces 4 haploid cells genetically different from the parent cells. The cell is diploid in the beginning, then the cell divides once becoming haploid due to the cells containing half the chromosomes. In prophase 1, the chromosomes condense and become more visible and each chromosome pairs with its homologue and allows crossing over to occur. One pair of chromosomes in the homologue is paternal, which is red, and the other pair of chromosomes is maternal, which is yellow. Crossing over is when the genetic information between homologous chromosomes that are paired together is switched. This creates the daughter cells to be genetically different. Also, during prophase 1, spindle fibers start to form. In metaphase 1, the pairs of homologous chromosomes line up across the metaphase plate. Together, the chromatids from a homologue attach to one kinetochore microtubule from one pole, and the chromatids from the other homologue attach to the other opposite pole of microtubules. In anaphase 1, the homologues move along the spindle apparatus to opposite poles of the cell. In telophase 1, a complete haploid set of duplicated chromosomes is in each half of the cell. The cell is ready to divide its cytoplasm in a process called cytokinesis. In cytokinesis, a cleavage furrow is started to create the completion of the first division of the cell. Once the 2 new daughter cells are created, it is time for meiosis 2. No chromosome duplication occurs between meiosis 1 and meiosis 2. Meiosis 2 is very similar to the process of mitosis. Remember that meiosis 2 is happening in 2 cells at once. Then meiosis 2 begins. In prophase 2, the spindle apparatus forms again and the chromosomes still are composed of 2 chromatids connected at the centromere. In metaphase 2, chromosomes are lined up on the metaphase plate, like in mitosis. The kinetochores of sister chromatids attach to microtubules that extend to opposite poles of the cells. In anaphase 2, sister chromatids are separated and move along the spindle apparatus to opposite poles. In telophase 2, the nuclei form in the 4 cells and cytokinesis occurs and completes the division, making 4 daughter cells with a haploid set of chromosomes that are genetically different from the parent cell.
We are never, ever, ever getting